Hello, my name is Helga Stopper. I'm a professor for toxicology at the Department of Pharmacology and Toxicology at Würzburg University in Germany. This short lecture is about the in vitro micronucleus test, its background and its application in genotoxicity testing. This lecture consists of a short introduction, then followed by some questions with literature recommendations for interactive learning, and then finally the answers for the questions. Micronuclei, what are they? Micronuclei are formed from acentric chromosome fragments or lagging whole chromosomes. They appear after completion of mitosis, and if the fragment or whole chromosome has not been distributed to the nuclear area of the daughter cells properly. Micronucleus formation can thus happen as a result of chromatin damage or breakage in formation of fragments or as a result of missing spindle attachment or spindle malfunction in the case of whole chromosomes. Micronucleus structures are enclosed in a membrane and are located within the cytoplasm of interphase cells. They are detectable upon staining of the cellular DNA, as you can see in the photo on this slide. Photo shows two nuclei and a micronucleus. They are stained with bisbenzimide 33258, a common DNA stain. So, what can micronuclei be used for? Micronucleus formation is an endpoint in genotoxicity testing, and genotoxicity testing is required for submission of material for approval of substances, for example, approval of chemicals or pharmaceuticals. But it is also an important endpoint for research regarding mechanisms of genomic damage induction in humans or wildlife biomonitoring. This lecture focuses on in vitro application of the micronucleus test in mammalian cells, but other options using micronuclei as endpoint are available. For example, mutagenicity testing of chemicals in vivo can be performed with rodents, usually rats or mice. There's the HANS micronucleus test. Micronucleus frequencies can be analyzed in environmentally burdened plants or animals. And it is possible to analyze micronucleus frequencies in human cells, such as peripheral lymphocytes or buccal mucosa cells. And the choice of the cell type can be related to the exposure, or it can be based on diseases. Literature recommendations for each of these options will be provided on the next slide. Here you can find some literature recommendations that will show the diverse application options for the micronucleus endpoint. Now, these literature recommendations are not mandatory for answering the questions that I will pose later. Instead, they're thought to help people along who want to use some of the variations of micronuclei that were listed in the previous slide. As I said, the focus of this lecture is mutagenicity testing of chemicals in vitro. And the protocols that are used for that are usually based on the OECD guideline test number 487, in vitro mammalian cell micronucleus test. In the micronucleus test in vitro, the substance cytocalcium B is often used. It is a cytokinesis inhibitor that inhibits the separation of the daughter cells after mitosis, but not of the daughter cell nuclei. As a result, binucleated cells are formed which may or may not harbor a micronucleus. They represent the cell population which has gone through mitosis since beginning of the substance exposure. The graphs show the separation of daughter cells after mitosis, labeled with 1, and the formation of a binucleated cell with the application of cytocalcium B, labeled as 2. The photo shows examples of binucleated cells with 1 or 2 micronuclei. Apoptosis is to some extent also detectable in the in vitro micronucleus test. If suspension cultured cells are used and slides are prepared with the use of a cytocentrifuge, apoptotic cells are visible on the slides. If adherent cells are used and the cell layer is washed before harvesting, apoptotic cells in the supernatant may be lost. If there are many cells in the supernatant, a preparation of control slides from the supernatant can reveal the presence of apoptotic cells. The photographs show you what apoptotic cells on such a slide look like. They have very condensed DNA, which have a similar shape and size of micronuclei, but just they are stained more intensely. The presence of many apoptotic cells on the slides may make it impossible to count micronuclei because their appearance is somewhat similar. A limited number of apoptotic cells may provide hints regarding the mechanism of action of the chemical. The relationship between micronucleus formation and apoptosis is not clear. Micronuclei may be a first step of the apoptotic process under some conditions or in some cell types, or it may represent a cellular decision against survival in the presence of damaged DNA if a cell goes into apoptosis. The following questions are relevant for the understanding of the micronucleus test in vitro, and they can be answered by looking at the reading material listed below. So the questions are: which cell lines can be used for in vitro micronucleus assay? How can toxicity be assessed, and why is that important? How is it possible to detect angiogenic effects, meaning the loss of whole chromosomes? What are recommended positive controls? Which chemicals and why? What could be the possible fate of a micronucleated cell, and why does that matter? The reading material consists of two open access publications. One is the OECD guideline, test number 487, and the other one is a publication by Gomez et al. For interactive learning, you must pause here for reading of the literature to answer the questions. The following slides will provide the answers to the questions. First question: which cell lines can be used for the in vitro micronucleus assay? For genotoxicity testing, a variety of cell lines or primary cell cultures can be used. Examples are TK6 human lymphoblastoid cells, L5178Y mouse lymphoma cells, V79 Chinese hamster cells, HepG2 human liver cancer cell lines, peripheral blood human lymphocytes, etc. The chromosomal stability of the cell line should be known, and cell lines with a low or stable micronuclei frequency are recommended. Usually the addition of a metabolizing system, so-called S9 mix, is required, unless the cells have metabolic capacity for the test item. For basic research, almost any cell type can be used, as long as the nucleus shape allows for analysis of micronuclei which are clearly separated from the main nucleus. How can toxicity be assessed, and why is that important? Micronuclei are expressed from existing damage after mitosis. Therefore, cells need to be able to go through mitosis. Otherwise, genomic damage induction may remain undetected. The fact that the cells are able to go through mitosis has to be demonstrated by the formation of binucleated and multinucleated cells, or by assessment of cell proliferation. The assessment of the cytokinases block proliferation index, CBPI, is a very good way to determine proliferation capacity. Numbers of mononucleated, binucleated, and multinucleated cells are counted, and the CBPI is calculated. CBPI is the number of mononucleate cells plus 2 times the number of binucleate cells plus 3 times the number of multinucleate cells, divided by the total number of cells. 2 examples are also given on the slide. With a high CBPI of 2.1, you have, for example, 100 mononucleate, 700 binucleate, and 200 multinucleated cells. And with a lower CBPI of 1.6, you might have 500 mononucleated cells, 400 binucleated cells, and 300 multinucleated cells. So if the control of your experiment has a CBPI of, for example, 2.1 or similar, that means there's a very good proliferation. A CVPI of 1.6, for example, in one of the treated culture, would show a reduced proliferation after substance treatment. How is it possible to detect angiogenic effects, which means the loss of whole chromosomes into micronuclei? Staining of kinetochores with antibodies or the centromeric DNA region with fluorescence in situ hybridization reveals the presence of the centromeric chromosomal region within the micronucleus. This could be due to the presence of a centromeric chromosomal fragment or the whole chromosome. If most of the induced micronuclei contain a signal, the test compound induces loss of whole chromosomes, usually due to mitotic disturbance. For further support of the idea of whole chromosomes being in the micronuclei, telomeres can also additionally be stained. The photos show two micronuclei without kinetochore signal on the left side and one micronucleus with a kinetochore signal on the right side. For example, between 20 and 50% of control cell micronuclei may contain a signal, while more than 90% of micronuclei in cells treated with a spindle formation inhibitor like colzimide or vinblastine may harbor a signal. What are recommended positive controls? Which chemicals and why are they used? Chemicals with known clastogenic or angiogenic capacity should be chosen, depending on the type of genotoxic mechanism expected from the test chemical. If metabolic activation is needed, because the test item is negative without metabolic activation, a chemical also requiring metabolic activation is a suitable positive control. Recommended positive controls are listed in the table, and for clastogenic activity without metabolic activation, we have methylmethane sulfonate, mitomycin C, or nitroquinoline N-oxide, or cytosine arabinoside. For clastogens which require metabolic activation, benzoylpyrene or cyclophosphamide are usually used. And as a positive control for angiogens, colchicine or vinblastine are good choices. What could be the possible fate of a micronucleated cell, and why does that matter? Micronucleated cells may die or survive, and may be mutated or be able to reintegrate the micronuclear material without loss of function. This may depend on the cell type. The cellular fate is important for the interpretation of the relevance of this endpoint. For example, dead cells do not form a tumor, while mutated cells, for example stem cells, may do so. Chromotrypsis is important in this context because it may lead to reintegration of rearranged chromatin into the nucleus, yielding chromosomes harboring many mutations. As a take-home message, I would like to state the following points. Micronuclei are an excellent and easy-to-assess endpoint for chromosomal and genomic mutation. The endpoint can be used in human biomonitoring and genotoxicity testing, as well as in ecotoxicology for the analysis of genomic damage in plants and animals. It is becoming more clear that the micronucleated cell itself can be involved in the carcinogenic process. A deeper knowledge about the fate of micronuclei and micronucleated cells might in the future also help to understand processes of chemical carcinogenesis even better. I would like to thank you for participating in this activity.